Think about what is happening inside your head right this very second.
Yeah, there's a lot going on.
Right. You're listening to my voice, processing the words, and maybe, visualizing the concepts. And if you had to picture what that looks like physically, you're probably imagining this massive glowing network of wires, like billions of neurons firing electrical signals back and forth.
Exactly, the classic wiring diagram.
Right. And for the longest time, that was the complete picture of how we form thoughts, memories, and movements.
Okay.
But what if there is a secret, hidden second internet operating right alongside those wires?
Oh, it's fascinating. An entirely parallel communication grid.
Yeah, a grid that has been practically invisible to us until incredibly recently.
It completely upends the traditional dogma of neuroscience, honestly. Because, for decades, the foundational belief has been that neurons, with their long, wire-like axons, are the primary communicators across the brain.
Right, they are the stars of the show.
Exactly, and everything else was secondary.
Yeah, and the other cells in the brain, specifically a type of cell called astrocytes, they were kind of brushed off as the supporting cast. The word glia literally comes from the Greek word for glue. You know?
Literally just glue.
Right. People thought astrocytes were just this structural mortar holding the genius neurons in place.
That was the assumption for so long. But recent work has revealed a much, much more active role. Astrocytes actually communicate with one another using physical tunnels called gap junctions.
Gap junctions, okay.
Yeah, and these connections aren't just for show. If you disrupt these gap junctions, you see major problems. Memory formation gets interrupted. The brain struggles to adapt to new information, a process called synaptic plasticity, and even the critical periods of visual and motor development in young brains can be, thrown completely off track.
Okay, wait. So if this secondary internet is so vital to basic brain function, why is it only being mapped out now? It feels like, something we should have noticed a long time ago.
You'd think so, right? But the delay comes down to a fundamental physical limitation in how scientists have historically studied brain tissue.
Oh, the tools weren't good enough.
Well, the methodology itself was the problem. If you want to understand how a brain works at a cellular level, the standard method for a century has been to take a brain, typically from a mouse, and slice it incredibly thin so you can put it under a microscope.
Which, sounds completely logical. You have to open the hood to see the engine.
It is logical, but it creates a massive blind spot. Because these astrocyte gap junction networks are microscopically delicate, the absolute second you push a blade through the tissue to make that slice, you physically sever the network.
Oh, wow. So you're breaking the very thing you're trying to look at.
Exactly. You introduce tissue damage artifacts, the cells react to the injury, and the natural communication just shuts down or distorts. To actually see how these cells share resources in real time, you can't look at a dead, sliced brain. You need to observe an intact, living brain.
Which is exactly the monumental challenge tackled by the research we are diving into today. And this is our mission for today's deep dive. We're looking at a groundbreaking 2026 paper published in Nature by a team of researchers including Cooper, Littleow, Chow, and their colleagues.
A phenomenal team, yeah.
Absolutely. The paper is titled Astrocytes Connect Specific Brain Regions Through Plastic Networks. And our goal today is to go through this brilliantly complex research exhaustively. We are going to unpack every single clever mechanism, every detail, and understand exactly how they finally illuminated this hidden grid without breaking it.
It's an incredible engineering feat, honestly, because to map a delicate microscopic network inside a living, breathing animal, you simply can't rely on physical tools. They had to get creative on a molecular level.
Right.
They needed to engineer a biological spy, essentially a vector-based Trojan horse that could sneak into the system and report back on the network's layout.
This is the part of the paper that absolutely blew my mind. The engineering here is wild. They built a custom viral vector to infiltrate the astrocytes. And in the paper, it's called, get ready for this, AAV5-GFABC1D-CK43.TIDHA.
It's quite the mouthful.
Yeah, let's be honest. That sounds like a terribly complex Wi-Fi password. Let's translate that alphabet soup into plain English, because every piece of that code does something super specific, right?
Right. It's essentially an algorithm. So the first crucial component is the promoter, the GFAT ABC1D section.
Okay, the promoter.
Yeah, think of a promoter as a highly exclusive VIP pass. When you inject a virus into the brain, it has the potential to enter neurons, blood vessels, immune cells, basically everything.
Right, you don't want it everywhere.
Exactly. This specific shortened promoter ensures that the genetic payload only activates if it finds itself inside an astrocyte. If it enters a neuron, it just sits there completely dormant.
Perfect. So our spy is selectively operating only inside the astrocyte.
Okay.
What's next?
The next part of the sequence is CUC43, which is encoded by the GJ1 gene. This is the primary gap junction protein found in astrocytes.
Okay, so if we imagine the astrocyte as a room in a massive office building, CUC43 is the literal tunnel door built into the wall that connects to the room next door.
That's a great way to visualize it. And now we get to the really innovative part, which is the turboid, or TID.
Ah, the TID part of the password.
Right. This is a highly active promiscuous enzyme whose only job is to attach a molecule called biotin onto anything that gets near it. And the researchers genetically fused this turboid enzyme directly to that CUC43 tunnel door.
I love visualizing this mechanism. It's so clever. I picture it like an aggressive hand stamper working the door at a nightclub.
That's exactly what it's like.
Right. So the infected astrocyte builds a tunnel to CA53, and it posts this bouncer, the turboid, right at the entrance. And the bouncer's only job is to furiously stamp anything that walks past it with molecular ink, which in this case is biotin. So, as natural molecules, ions, and nutrients pass through the tunnel from the infected cell into the neighboring uninfected cell, the bouncer stamps them. The next cell starts filling up with biotin ink, which gives us physical, traceable proof that the two cells are actively sharing materials.
The elegance of that system is just incredible. And to complete the spy kit, we have the final piece of the code, which is the HA tag.
The HA tag, okay.
This is simply a molecular flag that stays planted on the originally infected astrocyte. It basically allows the researchers to look at the brain later and easily identify patient 0, the cell that started the whole stamping process.
But wait, stop right there. Because if I'm thinking about the scale of a single cell, there's a lot of stuff floating around inside. How do they know the bouncer's standing in exactly the right spot?
That's a very valid concern.
Because if the enzyme is sitting too far inside the tunnel, it's going to clog up the traffic. And if it's too far away from the door, it's just going to start stamping random garbage floating around the cell's cytoplasm, giving us false positives.
Right. And the researchers knew that was a risk, so they had to measure the exact placement of this biological machine. To do this, they used a technique called expansion microscopy.
Expansion microscopy.
Yeah, they literally infused the brain tissue with a swellable polymer and expanded the astrocyte cells 8.7 times their normal size.
Wow. They literally blew them up like balloons.
Basically, yeah. Then, using super-resolution imaging, they measured the physical distance from the opening of the CUC43 tunnel to the HA tag on the enzyme.
And the measurement was exactly 20.73 angstroms. I mean, we are talking about 10 billionths of a meter. That scale is almost impossible to comprehend.
It really is. To put it in perspective, the actual opening of the gap junction tunnel is about 15 angstroms wide.
Okay.
So by proving the enzyme sits exactly at 20.73 angstroms, they demonstrated that the bouncer is positioned perfectly, just millimeters, well, molecularly speaking, outside the vestibule.
Oh, I see.
It's close enough to stamp molecules actively staging to pass through the tunnel, but far enough away that it doesn't block the physical flow of traffic.
That is staggering precision. Okay, let me just pause here and make sure I'm visualizing this entire setup correctly before we look at the results. Time for a quick recap.
Sounds good.
We know astrocytes communicate via these delicate C/43 gap junctions. Slicing the brain breaks them. So the researchers engineered a virus that forces specific astrocytes to build new tunnel doors equipped with an enzyme bouncer. That bouncer stamps any passing molecules with biotin, allowing us to map the network of sharing cells without taking a scalpel to the living tissue.
You've got the exact mechanism down perfectly.
Awesome. Which brings up a fascinating thought experiment for you listening at home. Imagine if you could drop a tracker on every single resource you share with your neighbors. The cup of sugar, the borrowed lawnmower, your Wi-Fi password. How far do you think your local personal network actually extends?
You'd probably be surprised by the reach.
Yeah, definitely. So now that we have the tool, what does this network actually look like when it's running in a living brain?
Well, the methodology here required immense patience. They took 3-month-old C57BL6 mice, and injected this viral tracer unilaterally, meaning into just one side of the brain and only into very specific targeted regions.
Right.
Then they simply let the mice live their normal lives for 3 whole weeks.
Wait, what, 3 weeks? Why not look immediately?
Because the delay is essential. It gives the viral payload time to express the fusion proteins. The astrocytes need those weeks to actually manufacture the new bouncer-equipped tunnel doors and organically integrate them into their naturally occurring gap junctions.
So the infrastructure has to be built first.
Exactly. And then comes the magic ink. For 1 week, they just give the mice biotin dissolved in their normal drinking water.
Okay.
The biotin circulates through the blood, enters the brain, and gives that turboid bouncer the ammunition it needs to start stamping the passing traffic. After a week of stamping, they finally look at the brain.
They didn't slice it, right? Because that was the whole problem we started with.
Right, slicing would ruin the 3D architecture. Instead, they had to make the entire brain completely transparent.
Transparent brain. Cool.
Yeah, they used a tissue clearing method called iDisco, and also another one called EdiboClear. Brain tissue is normally opaque, mostly because of the lipids, the fats.
Oh, okay.
So iDisco essentially strips away all the opaque fats while leaving the proteins like our HA tags and biotin ink perfectly intact in their original positions. Then they use light sheet microscopy to scan the transparent brain and generate these stunning, glowing 3D maps of the networks.
And the results from those 3D maps completely shattered older assumptions. Because historically, even when people admitted astrocytes shared things, they thought it worked like dropping water onto a wet sponge.
Yeah, just soaking outward.
Right. They assumed molecules just diffused outward randomly in a big uniform circle.
The imaging proved the exact opposite. Astrocytes do not operate like a random sponge at all. They form incredibly deliberate, highly specific routes.
We're really specific.
Yeah. For example, when they injected the tracer into the motor cortex, the part of the brain controlling movement, the resulting astrocyte network stayed highly exclusive to itself. It didn't carelessly bleed over into surrounding functional areas.
But it wasn't isolated everywhere, right?
Right.
Because when they injected the prefrontal cortex, the area for complex decision-making, or the hypothalamus, those two networks heavily overlapped.
Yes.
Which suggests those regions are engaging in bidirectional, continuous communication along the exact same astrocyte highways.
We also started seeing massive central hubs. The superior colliculus, for instance, acts as a robust bilateral hub connecting both hemispheres of the brain. But what's truly wild is that the architecture is asymmetrical.
Asymmetrical?
Yeah, if you look at the network on the right side of the brain, the projection to the left side isn't just a perfect mirror reflection. The contralateral side has its own unique, distinct structural shape.
Wow. I'm trying to picture the shapes of these highways. Are they just like solid blocks of glowing cells?
Far from it. The geometry is incredibly varied. Sometimes they form continuous, unbroken chains that physically traverse the white matter tracks, like the corpus callosum, seemingly following the paths of specific neuronal axons.
Yeah.
In other brain regions, they form intricate honeycomb patterns. And these honeycombs are highly selective. They will deliberately connect to certain distant astrocytes while completely bypassing and excluding other astrocytes sitting right next to them.
That is so deliberate.
It is. They even form vertical columns, connecting the deepest parenchymal tissues of the brain right up to the outermost surface layer, the glia limitans superficialis.
Okay, I have to channel my inner skeptical peer reviewer for a second here.
Go for it.
We gave these mice biotin in their drinking water. Biotin goes into the stomach, into the bloodstream, and travels through the blood vessels into the brain. How do we know for absolute certain that this glowing map is actually a cellular network? What if the biotin is just leaking out of the blood vessels, drifting around the brain tissue, and getting absorbed randomly by nearby astrocytes?
It's the most important question to ask, and the researchers absolutely anticipated it. To prove this was strictly a gap junction phenomenon, they ran a conditional knockout experiment, or CKO.
Okay, what did they knock out?
They bred specially modified mice that lacked both the GJ1 gene, which codes for the main CGAPS43 door, and the GJB6 gene, which codes for a secondary tunnel called CGI30.
So they basically went in and boarded up all the doors between the astrocytes.
Exactly that. Using a tamoxifen trigger, they selectively deleted these gap junctions only in the adult astrocytes. And when they ran the exact same viral tracer and biotin water experiment in these knockout mice, the massive brain-wide networks completely vanished.
Wow.
The glowing biotin signal shrank down to only the single originally infected cells.
And just to completely put the nail in the coffin of the blood vessel leak theory,
They stained the blood vessels themselves with a marker called tomatolectin. When they layered that over the 3D maps of normal mice, there was zero correlation. There was no leakage or spreading of the biotin signal along the vasculature. The vast distribution relies entirely on those specific astrocyte gap junctions.
That's just brilliant experimental design. Okay, time for another quick bullet recap before we move to the next part.
Let's hear it.
So we've learned these networks are not random diffusions. They are highly specific, complex highways taking the shape of chains and honeycombs. And we know for a fact, thanks to the knockout mice and the tomato lectin stain, that these highways are completely dependent on the gap junctions. No doors, no network.
Which brings up a fascinating conceptual point about the brain's overall design.
Yes, because if these astrocyte networks don't perfectly overlap with the neuronal networks, it begs a question for you listening. If they are different, what distinct parallel function are they serving in your brain right now? They are clearly sending something entirely different along completely different routes than the electrical signals of your neurons.
And that observation leads us to arguably the most important question of the whole study. We have the map. We know the roads are structurally specific. But are they permanent?
Right, are they static?
Are these pathways like poured concrete roads laid down during early brain development, or are they plastic? Do they change shape based on what you actually experience?
To figure out if the roads can change, the researchers turned to a classic neuroscience case study, the barrel cortex.
The barrel cortex is the region in a mouse's brain that processes sensory information from its whiskers, and whiskers are highly sensitive, essential tools for a mouse.
Super important for them.
Yeah, and we've known for a long time that if you trim a mouse's whiskers, depriving it of that sensory input, the electrical neurons in that specific brain region will physically rewire themselves to adapt to the loss.
It's a classic use it or lose it adaptation. The brain says, I'm not getting signals from these whiskers anymore. Let's reallocate the hardware to something else.
Exactly.
So, to test the astrocytes, the researchers trimmed the whiskers on one side of a mouse's face for 28 days to trigger this remodeling. Then they injected a viral mixture into the barrel cortex. But it wasn't just our astrocyte tracer this time. They co-injected a neuronal marker driven by the CAM-KII alpha promoter, which specifically lights up excitatory neurons.
That dual labeling is vital. By using two different markers, they could essentially turn on the GPS for both internets simultaneously. They could watch the neuronal wires and the astrocyte highways in the exact same living brain. They kept the whiskers trimmed for another 4 weeks, and then they looked at the maps.
And the results were dramatic. The astrocyte networks physically shrank.
Vastly. In a normal, naive mouse with intact whiskers, the barrel cortex astrocytes project a robust, long-range network all the way out to the prefrontal cortex. But after a period of sensory deprivation, those specific long-range connections completely disappeared.
Well, just gone.
The road was simply gone. Meanwhile, connections to the midbrain remained stable.
This highlights such a staggering divergence from the old dogma. Because we used to assume astrocytes just wrapped around whatever the neurons were doing, basically following their lead. But while some local connections are shared by both systems, many of the long neuronal projections they traced had absolutely zero corresponding astrocyte connectivity.
They are completely distinct structural infrastructures. The two internets are operating independently.
But wait, there is one incredibly rare exception found in the data that we absolutely have to talk about. While neurons and astrocytes generally stay strictly in their own lanes, the researchers found evidence that they sometimes cross over.
Yeah, this was a tiny detail in the paper, but the implications are massive. Let's explain the connexin mismatch because it relies on some basic structural biology.
Yeah.
Gap junctions only connect to compatible types. It's like trying to plug a USB-C cable into an old HDMI port. If the shapes don't match, the connection fails.
A good analogy.
Neurons typically use a gap junction protein called connexin 36, or C-by36, which is classified as a delta type. Astrocytes primarily use CX43, an alpha type, and C-by30, a beta type.
Okay, so different types.
Right, and delta types physically cannot dock with alpha or beta types. Therefore, the biotin ink being stamped inside an astrocyte should never, under any circumstances, end up inside a neuron.
And here's the crazy part, there is an exception to the rule.
There is. When they injected the tracer into the motor cortex and followed the highways down into the spinal cord, they found biotin ink inside motor neurons.
Meaning the astrocyte internet actually found a compatible port and plugged directly into the neuronal internet.
The only way that is biologically possible is if those adult motor neurons are expressing compatible alpha-type C-Guy43 gap junctions.
Which is huge.
Right. Previous literature has hinted that motor neurons might do this during early development or maybe after a severe spinal injury, but finding this direct astrocyte to neuron communication in a healthy adult mouse implies that these two internets directly interface during normal motor control far more than we ever realized.
Okay, my final mind-blown moment on this topic is the speed at which all of this happens. Because when we talk about infrastructure remodeling, like shrinking an entire biological highway to the prefrontal cortex, you might think a process like that takes months or years.
It seems like it should.
But the half-life of a single C/43 gap junction protein is estimated to be between 1.5 and 5 hours.
The turnover rate is just breathtaking. It means this entire brain-wide infrastructure of tunnels and connections is constantly dissolving and rebuilding itself. Day in and day out, hour by hour, the network is shifting to meet the metabolic and functional demands of the brain.
Okay, last quick recap for you guys. Sensory deprivation, like whisker trimming, physically shrinks these astrocyte networks independently of neurons. And with a rapid C/43 half-life of 1.5 to 5 hours, this network is constantly remodeling itself.
Yep.
So consider what your brain is doing right now. When you learn a complex new skill like a new language, a musical instrument, or even just absorbing the dense information in this deep dive, you are literally building new biological highways in real time. Your astrocytes are constructing tunnels and laying down infrastructure while you sleep.
It forces a profound shift in how we visualize the mind.
So bringing this all together, what does this ultimately mean for our understanding of the brain? This research completely rewrites the architectural blueprint. We are no longer just looking at a static wiring diagram of electrical neurons.
Far from it.
We are looking at a highly specific, structurally plastic, and incredibly dynamic secondary internet. These interconnected astrocytes are buffering ions, distributing essential metabolic resources, and occupying distinct functional niches completely independently of the neurons they surround.
Which leaves us with a massive lingering mystery. The viral vector we discussed, that bouncer at the door, it is promiscuous. It stamps absolutely anything that passes through the gap junction.
Right.
So thanks to this tool, we finally have a beautiful, high-definition 3D map of the highways. But what we don't know yet is what exactly is driving on them.
We can see the roads, but we don't know what the cargo is.
That is the next frontier. What are the specific metabolic resources, the ions, the complex signaling molecules physically passing through this hidden internet?
It could be anything.
Are these cells sending distress signals during a stroke? Are they delivering extra energy reserves when you're deeply focused on a hard problem?
Yeah.
How does the traffic on these incredibly plastic highways change as we age, or when the brain experiences neurodegenerative diseases like Alzheimer's?
Mapping the roads was the first, impossible step. Intercepting the actual messages is next. Thank you so much for joining us on this incredibly detailed journey through the hidden architecture of the mind. The next time you picture the brain inside your head, don't just see the electrical wires. Remember the second internet, silently and constantly rebuilding itself hour by hour, keeping the whole incredible system alive. Stay insanely curious.